Pharmapal.
Drug-receptor interactions, nonspecific versus specific activity. Hey everyone! Welcome back to our channel. Today, we're diving into a fascinating topic in medicinal chemistry, structurally nonspecific and specific activity. If you've ever wondered how drugs interact with our bodies and how their structures influence their effects, you're in the right place. So, let's break it down together. First up, let's talk about structurally nonspecific activity. This refers to drugs that don't have a specific target in the body. Instead, they exert their effects based on their physical properties, like solubility and partition coefficients. Examples of these include amides, ethers, ketones, and chlorinated hydrocarbons. These substances can accumulate in lipid-rich areas of cells, leading to their narcotic effects. Think of them as general anesthetics or certain insecticides that affect many systems in the body rather than just one specific receptor. Now, let's shift gears to structurally specific activity. This is where things get a bit more intricate. Here, the activity of the drug is closely tied to its molecular structure. Factors like functional groups, the shape of the molecule, and even the distance between certain atoms play a crucial role. For instance, a drug might bind to a specific receptor in the body, like how cholinergic drugs interact with acetylcholine receptors. The unique structure of each drug determines how well it fits into its target receptor, much like a key fits into a lock. Speaking of receptors, let's talk about drug-receptor interactions. A receptor is essentially the site in our body where a drug exerts its effects. It's like a regulatory hub for physiological processes. When a drug binds to a receptor and triggers a response, we call it an agonist. On the other hand, if it binds but doesn't activate the receptor, it's known as an antagonist, blocking the receptor's activity. Now, let's discuss affinity. This term describes how well a drug binds to its receptor. Receptors are dynamic and have specific requirements for binding, which means that even small changes in a drug's structure can significantly alter its affinity and, consequently, its effectiveness. This brings us to stereochemistry, the three-dimensional arrangement of atoms in a molecule. It's crucial because different isomers can have vastly different biological activities. For example, one optical isomer might be a potent pain reliever, while its counterpart might not have any effect at all. Another important concept is the quantitative structure-activity relationship, or QSAR. This is a method that helps predict how changes in a drug's structure can affect its biological activity. It's like having a roadmap that guides medicinal chemists in designing new drugs. And let's not forget about acidity and basicity. The acidic or basic nature of a drug can significantly impact its solubility and how it behaves in the body. For example, carboxylic acids and amines are common functional groups that influence drug behavior. Next, let's discuss drug-receptor interactions. So, let's kick things off by understanding what a drug-receptor interaction actually is. In simple terms, a receptor is a specific site in our body, usually a protein, where drugs bind to exert their effects. Think of receptors as the locks and drugs as the keys. When the right key fits into the lock, it opens the door to a biological response. Now, why is this interaction so important? Well, most drugs work by binding to these receptors, which can be found on the surface of cells or even inside them. This binding triggers a series of reactions that lead to the desired therapeutic effects. There are two main types of drug-receptor interactions, specific and nonspecific. Let's start with specific interactions. These occur when a drug binds to a particular receptor, leading to a precise biological response. For example, let's talk about cholinergic drugs. These interact specifically with acetylcholine receptors, which play a crucial role in muscle movement and memory. On the other hand, we have nonspecific interactions. These don't target a specific receptor but rather affect various physiological processes. For instance, some general anesthetics can interact with multiple receptors, leading to sedation without a precise target. Now, let's discuss two key terms, affinity and efficacy. Affinity refers to how well a drug binds to its receptor. A drug with high affinity will bind more tightly to the receptor, while one with low affinity may not bind as effectively. Efficacy, on the other hand, is about how well that drug activates the receptor once it's bound. A drug can have high affinity but low efficacy, meaning it binds well but doesn't trigger a strong response. Speaking of efficacy, this brings us to agonists and antagonists. Agonists are drugs that bind to receptors and activate them, producing a biological response. Think of them as the activators. Antagonists, on the other hand, bind to the receptor but don't activate it. Instead, they block the receptor from being activated by other substances. You can think of them as the blockers. Now, several factors can influence these interactions. The concentration of the drug, the presence of other competing substances, and even the physical and chemical properties of the drug itself can all play a significant role. It's also important to note that receptors are dynamic. They can change their shape or concentration in response to various factors, which can affect how drugs interact with them. In summary, drug-receptor interactions are fundamental to pharmacology. Understanding how drugs bind to receptors helps us develop better medications with fewer side effects. Thanks for joining us today. If you found this video helpful, don't forget to like, subscribe, and hit that notification bell for more exciting content on medicinal chemistry and pharmacology. Until next time, stay curious and keep learning.
Pharmapal.